All right, in this video, we're going to talk a little bit about human genetic diseases and inheritance. So, in studying some organisms like peas and fruit flies, you can do controlled crosses. They have short generation times. They produce lots of offspring. Humans have the exact opposite. We can't really tell people who they can mate with. Our generation times are felt fairly long. They live about as long as the person studying them. And there are a few offspring produced each generation. You don't have, you know, large families of certainly not more than a couple kids for most families. So to get around that problem, we use a technique called pedigree analysis. And pedigree analysis has shown Mendelian inheritance patterns in human inheritance as well. Things like traits like having a widow's peak or not, attached and free earlobes. We can basically make a data map on a family tree by adding the genotypes and looking at phenotypes with the family tree. So here's an example. In pedigree analysis, there's a couple things you need to know. Squares are males, circles are females. If it's filled in, that has the trait we're studying. If it's not filled in, then it doesn't have the trait we're studying. So what's the likely inheritance pattern here? Well, we see that both parents have the trait. Some of the children have the trait. Probably likely that this is a dominant trait. But we'll talk about how you figure that out later. So when you're looking at a pedigree, there are certain things you want to look for. One of the first questions you want to ask when you're studying a pedigree, are there about an equal number of males and females that are affected? If that's true, it's an autosomal trait. If you have more males than females, it's likely a sex-linked trait. Does every affected individual have at least one affected parent? That's a dominant trait. Does the trait tend to skip? Now, it doesn't have necessarily always skip, but does it tend to skip generations? Then it's a recessive trait. So here's an example of an autosomal dominant trait and what we would see in the pedigree. You see every affected individual has at least one affected parent, and there's about an equal number of males and females. Certainly not more males than females. So this would be autosomal dominant inheritance. Here's an example of an autosomal recessive. Now, one of the things you see here is you see half filled in circles. Those represent carriers or heterozygous individuals. In autosomal recessive traits, again, in generation 2, you have several individuals that have the trait, but neither of their parents had the trait, so they must be carriers. Now, on an autosomal recessive trait, you're not necessarily going to have all the carriers marked because you may not know. So that's what that shows us. Here's an example of a sex-linked recessive trait. This is for factor VIII hemophilia. This is from two royal families. And you'll notice Queen Victoria is half filled in. Queen Victoria was the first person to have the mutation that led to factor VIII hemophilia, and one of her sons, Leopold, was the first individual. So if we look at this, you see a few female carriers, but you don't see any females with the trait hemophilia, but you do see some males. So that tells us it's a sex-linked recessive trait. So when do we use this? Well, we can use it just to study and look and see at inheritance patterns, but more commonly today, we might use this when we have a couple that has maybe a history of a disease in their family. They want to know, hey, if we have kids, what's the probability of those kids having this particular genetic disorder? So we can, determine, what's the chance of them having things like albinism, sickle cell disease, PKU, and so on. And again, some of those are things we can treat like PKU. Some of those are things we can't treat like cystic fibrosis and Tay-Sachs. Some are going to lead to premature death. Some are just going to mean that you got to be more careful. Genetic testing is something that is used oftentimes by genetic counselors, where we look at things like the cells from an embryo. We can do it through amniocentesis. We can do it through chorionic villi sampling. We grow those cells. We look at things like the chromosomes. We might do some testing to see if they might be an individual with a particular trait. Things like Down syndrome, we could see that on a karyotype. Other things we would have to do other testing to find out. Things like uh, restriction fragment length analysis, or what we commonly call DNA fingerprinting, sequencing genes to see if they have a particular mutation. So sometimes you have these recessive diseases because you have a malfunctioning protein, or no protein is being produced at all because of the mutation there. Heterozygous individuals are often referred to as carriers for these recessive diseases because they have one good copy that normally compensates for the bad copy. In the heterozygote crosses, again, you can see heterozygote crosses, you can have a 1 out of 4 chance of having that particular recessive disorder. With cystic fibrosis, this is a recessive disease, and we see for some recessive diseases, we see them in certain ethnic groups or people of certain ethnic backgrounds. Cystic fibrosis tends to affect primarily people of white European descent. In that group, 1 out of every 2,500 births results in a child with cystic fibrosis. 1 out of every 25 is a carrier. So this disease, there's a gene that codes for a memory protein that transports calcium ions across the cell membrane. If it's defective, it results in thicker, stickier mucus that coats the cells in the lungs, in the digestive tract, causing bacterial infections, difficulty breathing, pneumonia. Without treatment, the children are going to almost certainly die before they reach 5. With treatment, they can live past their late 20s, and we're getting even better at this, and people are living longer and longer with cystic fibrosis. You can see kind of how that works. One of the things that happens a lot of times with cystic fibrosis is that parents have to do percussion therapy where they basically pound on the kid's chest and back to loosen up that mucus a couple times a day. Tay-Sachs is another recessive disease that affects a certain group more likely, primarily Jews of Eastern European descent and Cajuns. 1 out of every 3,500 births results in Tay-Sachs, which is 100 times greater incidence than non-Jewish or non-Cajun groups. In this case, a lipid builds up, or fat builds up in the brain cells and destroys them because they're missing in the enzyme to break down that lipid. Usually you see they're okay at birth, but within a few months after birth, they start to show symptoms. Their developmental progress is delayed. They might have seizures. It might lead to blindness, degradation of muscles, mental performance. And this one usually results in the child dying before they really reach the age of about 5 years old. Sickle cell disease affects primarily Africans. Here in the United States, about 1 out of every 400 African Americans, which is a really high frequency. It's caused by a substitution of a single amino acid in the hemoglobin gene that carries oxygen, so it has anemia, but it also results in misshapen cells. We get this pleiotropy because those misshapen cells stick together, it damages organs, and leads usually to premature death. Now, the reason for sickle cell, we see, again, 2 alleles are co-dominant in this case. The normal and mutant allele results in some normal and some abnormal cells. Carriers are usually okay. Sickle cell can be triggered when there's oxygen stress, like during exercise. So a carrier may, normal day-to-day, they're fine, but if they exercise heavily, they may see the symptoms of sickle cell showing up. So why is this so prevalent in people from this group? Well, what we found is that heterozygous individuals for this trait have an advantage if they come from a part of the world, or they live in a part of the world where malaria is an endemic problem. Malaria is a parasite that spends part of its life cycle in red blood cells. In tropical Africa, malaria is very common, also in parts of South America. Homozygous individuals often die from malaria if they catch malaria. But again, we're getting better at treating this and preventing it. We have anti-malarial medications and things like that. But again, in poorer parts of the world, they may not have access to that. Homozygous recessive individuals typically die from sickle cell anemia. Heterozygous individuals usually are free of both. They're not usually going to have sickle cell unless they're stressed, and they're resistant to malaria because of the trait. So they had an advantage, which creates a higher frequency of the sickle cell allele. It means we have more people that are carriers, which ends up with more offspring that have the disease. Huntington's disease is actually a dominant trait, named after George Huntington. There's a repeated mutation on the end of chromosome 4. You get a bunch of these CAG repeats, which means you have a bunch of glutamine amino acids at the end of the protein. It was one of the first genes to be identified in the human genome. So there's this Huntington protein that builds up and causes damage to cells in the brain. Leads to memory loss, muscle tremors, loss of smooth controlled movement, which we call chorea. It used to be called Huntington's chorea. Starts usually until about 30 to 50. Everything's perfectly normal, but then starting somewhere between age 30 and 50, we start to see a progressive buildup of signs and symptoms of the disease. Leads to premature death. About 10 to 20 years after the signs and symptoms first show up, the person's typically going to die. Now, one thing, we do have a test for this. So if you have a history of this in your family, you might want to go to the doctor and say, hey, I want to get tested for this. And again, that brings up a question. Do you really want to know? If you're going to be normal for 30 to 50 years, do you want to really know that at some point you're going to start developing this disease? Some people might say, yeah, I want to know so I can do everything before that progresses. Some people might say, I don't want that hanging over me. And they might say, I don't really want to be tested because we can't do anything for it right now. So there's a genetic role or a cultural role in genetics as well. Question that a sociologist might ask is, why do all cultures have this taboo against incest? Why do we have laws and cultural taboos forbidding marriage between close relatives? That's pretty much a universal thing in all cultures and in most countries. And the reason for that, there's a genetic reason why that's a good thing. For a lot of the diseases we talked about, they were recessive diseases. That means you have to have parents who either have the disease or at least are carriers. So it's fairly unlikely that two unrelated carriers for the same rare harmful recessive allele will meet, mate, and have offspring. But if you have individuals that are close relatives, they're likely to have similar genetic makeup, so that there's an increase with what we call consanguous or same blood matings. Individuals who share a recent common ancestor are more likely to carry the same recessive alleles. And we saw this in the royal families of Europe back in those days when royal families were only allowed to marry other royals. That gene pool got very, very small. And so you tended to be marrying people that you were somewhat related to. And we saw these recessive diseases happening more often in individuals of these royal families. We see the same thing today in groups like the Amish, or people that typically only marry within their group.